There we go. So we are recording. Let me share my screen. Because if I remember correctly, you didn't make any mistakes that a lot of people didn't make. So this video should be very relevant to most people who do this lab. All right. So you can see my screen, correct?
Yeah, I can see it.
Perfect. All right. So we'll look at this. Okay, photosynthesis. So you got looks like you got all the pre-lab correct. Yep, full credit. Where are the comments? Here we go. Okay. So, hypothesis. What is your hypothesis for this question, and why do you believe it to be true? So, I believe the amount of pH present. Okay, yeah. So, let's just start back up a little bit. So, the question is, does the rate of photosynthesis increase as light intensity increases? So, basically, when you think about it, there's only three answers. Yes, it does, or two answers. Yes, it does, or no, it doesn't. Really, it's that simple. And then, of course, you have to say why, right? So you could say, yes, I believe the rate of photosynthesis increases as light intensity increases because photosynthesis relies on photons or something like that, some explanation. So here we have, I believe the amount of pH present and the conditions the plants are in will affect on the rate of photosynthesis and respiration. Okay, so there's a few issues with your hypothesis. One, you kind of got it flip-flopped. So you're saying that the conditions will affect photosynthesis. How do I put this? You were saying that the pH. Well, I guess we'll focus on that part first. To say that the pH would affect the photosynthesis and respiration, that's incorrect, right? Because that's the backwards. What we're saying here is the conditions. That part is correct. The conditions specifically, and you would need to say what, as light intensity increases, it'll affect photosynthesis, or as it decreases, it'll affect whichever it is you believe. Does that make sense? So, well, I guess what I'm trying to say here, the first thing I'm trying to say is the pH does not affect photosynthesis, right? Does that make sense?
Yeah, that makes sense.
Okay. The important thing here is that we are using pH to measure photosynthesis. So, in a way, this is the opposite of true, right? We would say photosynthesis would affect the pH. But again, that doesn't even matter because here we don't even need to talk about pH. We're just saying, you're just answering this question. Do you believe it to be true or not true? Or do you believe photosynthesis will increase as light intensity increases or not? So that's why you miss points on this one. You really don't really address the question. Does that make sense?
Yeah, that makes sense.
Okay. And another thing, a small thing, you wouldn't lose points for this. I was just pointing it out that we used algae, not plants, and of course, again, you didn't lose points for that in the lab, but that would be important for the lab report. That would be a mistake that would cost you some points in the lab report. So yes. Anyway, what was your hypothesis? I forget reading this, but now that you know what I mean when I'm asking for the hypothesis, like, do you think light intensity? Basically, do you think light intensity affects photosynthesis or not? What do you think?
I think it does affect photosynthesis.
There you go. All right. So, any questions about the hypothesis question?
Nope.
Okay. Let's take a look here. There's the table. Okay. I should point out, too, not just for you, but I guess for other people, too, that might be watching this. We did extra cuvettes. This is how it was written, the way it was here, which is exactly what was expected of you. So obviously you didn't lose any points. But some people did include that extra, like as if you were to like highlight this and then insert an extra one. Because remember, we had an extra cuvette. That would have been extra credit. And then let's see. What do we have here? So let's start here. They all started at 7.7, so that's nice and easy. And then that one ended at 9.1. So good. Then 8.5. Okay. And then I guess 7.5. Yep. Okay, good. All right. So your table looks good. Describe your results. Well, let me just look to see where I have comments. It looks like you did good on that. You did good on that. Anything that I didn't leave comments on, then you must have done properly. Okay, this is good. Yeah. So this time it looks like you really did get it for the experiment, right? What is your hypothesis and why do you believe it, basically? And you said, I believe that organisms will not respire with lack of light because it is overall energy sourced by the light. So it is able to convert it to its proper energy source. So yeah, you answered the question and you explained why you believed it to be true. And that was all that was required. And then there's your table. So that's good from 7.7. There's no comments on there. So you must not have made any mistakes. This was good. No mistakes there. Got a little extra credit here because you got specific. Okay. And then that all looks good. Okay. That all looks good. That all looks good. More extra credit. Oh, yeah. Interesting. So, did you mean the lab report perhaps? Because if we're looking at this, I don't know. I should have done this to begin with. You actually got 16 out of 15. So, you had extra credit. You did really good you did very well on this lab, points wise.
Both the lab and the lab report, because I thought like maybe the actual like lab reflected how I did on the lab report.
Gotcha. Okay. So let me take a look here. First of all, let me make sure it's properly recorded. It is. Okay. 5 and 15. Let's see. So what was it? Sorry to change the subject, but before we look at your lab report, which lab was it that you had me redo like for the you remember the last lab was just choosing an old lab to fix? Which lab did you have me do for that one? Do you remember?
Yeah, it was biological molecules.
Okay. Gotcha. Anyway, let's look at the lab report if you want. Let me pull this up. Instructions. Nope, not that. Go back here. Yeah, so yeah, your photosynthesis lab itself was very good. Like I said, we discussed the one issue that you had, and then with all the extra credit you got, you actually made up for that and then some. But let's pull up the lab report. Okay, where are we at? Lab report for synthesis, genetics. Here we go. Where are you at? All right, finally got it. Pull it up. Share my screen. All right, so can you see it? The lab report yet? Has it popped up?
Yeah, I can see it.
Perfect. Okay. So obviously you can look at here and see the exact points, but then the comments give you more feedback as opposed to just the points. You can see what might be wrong. All right. Yeah. So this wasn't this is a small mistake, especially considering that you fixed it literally 2 sentences later. But you said does different light affect the rate of photosynthesis? And basically, you just needed to be more specific, like you were here, right? Do different colors of light, because when we say different light, that can mean really we could do extra experiments, and I might do that in future semesters. That you've kind of inspired me. Maybe I'll do one where we have like, all right, well, here's an LED light versus an incandescent light or something like that. But anyway, yeah. So you had to be specific with that. So that's one issue. This issue, this isn't again not a big deal, but really you don't need to. I don't know how to explain it. You don't have to put your predictions or your hypotheses in here, right? So you wouldn't say in this case, there's no need to say the strongest color was supposed to be this or that, because really we don't know. That's the purpose of the experiment, is to figure out what the strongest color is, so to speak, right? So on one hand, you lost a point for including that in the abstract where it doesn't go. And also true, that's just also not a correct statement. What we do know from lecture is that the strongest color, depending on how you look at it, should be either white because it's all the colors, or it's blue because it's higher energy. But anyway, does that make sense? The biggest issue there, and it's you only lost 1 point for it, is that you included that in the abstract where it doesn't necessarily belong. Make sense?
Yeah, I see the mistake.
Perfect. Okay. There were a few minor errors that were taken into consideration. All right. And this is another one of those things where it just doesn't belong in the abstract. So that's good that you included it, but that goes in the discussion. Basically, the abstract is supposed to be pretty straightforward. Like, here's the question. Here's basically how we did it, which is right here. Well, again, not the exact instructions. So that portion was incorrect for you to say that. But luckily, you went on to not give the exact instructions. You did what you should do, which is a basic setup or a basic explanation of the setup. So you had the algae bead cuvettes under each color light. Um, technically, measure the pH levels, right? So that's good. You did a basic description of how you did it, and then of course the more important part, as far as points are concerned, is this right here. You needed to have a basic summary of the conclusions. So here's the question. Here's what we did, and then here's the results, right? So you would say something like, and then come to find out, white was the best color and green was the worst, or whatever the case may be. Make sense?
Yeah.
Okay. For the introduction, plants are able to use sunlight to synthesize foods from water and carbon dioxide. Oh, yeah, good. So that was a decent explanation of photosynthesis, especially considering that you included the thing about carbon dioxide, which will be important later. So that part was good, but you needed to also explain respiration, because later in that introduction, you need to thoroughly explain the pH thing. So in order to thoroughly explain the pH thing, then you need to also thoroughly explain photosynthesis and respiration, at the very least, how they use or produce carbon dioxide. Again, which you did a great job with the photosynthesis, just not the respiration. And again, I already brought this up, right? So does different light affect the rate of photosynthesis? Again, you just needed to be more specific. I believe the red colored light would be the strongest, and green being known as the weakest. Oh yeah. So in this case, again, you had your hypothesis, but you needed to explain why you believed it. You know what? Let me think about this. The color light source. Yeah, and yeah. In this case, hold on. That is the why. See, this is why I like to do these things. I'm giving you, delete this. I'm going to add a point because I just misread this. So this was your hypothesis, and then this was your reasoning why. So you get a point back. Let me go ahead and fix that right now. Otherwise, I will forget. There we go. See, this is why I like doing these things. Anyway, any questions so far?
Nope, no questions so far.
Okay, does the light? Okay, had that. My group members and I were able to assess the pH levels over the course of time. Okay, so this one, you just needed to explain it in more detail, right? So anybody who reads the lab report, the whole thing you should have enough information in the introduction where the whole thing makes sense. So here's where you would talk about all those things that you answered in the pre-lab, where you would say, as you said up here, well, I'll start with respiration. Respiration produces carbon dioxide. Carbon dioxide mixes with water, which makes an acid. Therefore, when things respire, the pH goes down because it becomes more acidic. Likewise, photosynthesis uses carbon dioxide, so it takes that out of the water, so things become less acidic, so the pH increases as things photosynthesize. Make sense?
Yeah, it makes sense.
Perfect. Okay, so that's what you needed in the introduction. That way, when the reader goes on to read the rest of your lab report, they'll understand what that means. Like, otherwise, they'll be like, "What in the world? What does the pH have to do with this? I thought this was about photosynthesis." Anyway, materials and methods should have been in paragraph form instead of a list, but that's no big deal. That's, I think, like 1 point lost, so that's no big deal. 4 cuvettes, algebra, water, pH indicator, good. Oh yeah, and I know it seems redundant, but you also need to mention the light, right? So we used 4 lamps, each with their own different color light bulb, something like that. And then again, that's probably only 1 point. And you did kind of hint at that later. 20 cents, and you didn't lose point for this, and I'm just now seeing it. But I would mention, like, not that you don't need to fix anything, but if you were to rewrite it, I would not even put the 20 centimeters. if I remember correctly, because we didn't actually measure 20 centimeters, right? Didn't we just measure to make sure that they were all equal? Anyway, that's so that would be make it more correct, but you didn't lose points for that. Let's see. Now it's time to start collecting data. Oh, and again, this should be first person past tense. So again, it's not like you're not supposed to copy it necessarily from the lab itself, but just kind of describing what you did as opposed to giving someone instructions on how to do it. But anyway, let's see. Within a 5-minute time mark, remove the cuvette from a designated place and give it a little mix. Should display a certain color, which will be measured using the trusty pH indicator. Oh yeah, so I was just pointing out for there, the pH indicator is the liquid that's in the cuvette. So yeah, like you said right there, there's water in there. There's also that liquid, the pH indicator. So you need to describe how you can use the color of that pH indicator to figure out the pH. In which case, you would say, refer to the pH strip, that little chart that people could compare it to. So the results, again, you just had some stuff in here that was supposed to be in the discussion, not the results. So that was just 1 point lost right there. This should have been in paragraph form instead of a bullet list because, essentially you're taking what was in the results, or what was in the table, and putting it into words. And if I remember correctly, I didn't take a point for that one. No, I didn't. So you didn't lose a point for that. But yeah, technically this should be just like I had you do it in the lab itself, right? You had the table, and then afterwards you had to put it into a sentence. So you would say something like, the cuvette in the green light started at a pH of 7.1 and ended at a pH of 8.5 with a total change of 1.4. Let's see. Color light have different strengths. So I would just point it out, not necessarily that you lost points for that, but it isn't necessarily the strength because we had a whole different experiment for that. Like you could talk about light intensity, and there was an experiment done for light intensity. So it's not necessarily the strength, but it's the ability of the chlorophyll to absorb that wavelength is what we focused on. Such as the green light source having exact results of change as the red light. Oh yeah, so let me focus on this. Then you wrote, but the errors had buffered any accurate results there could have possibly been, such as the green light having exact results. So what I was pointing out here is you're supposed to talk about the errors in the experimental design, right? So as opposed to the errors in the results. Now, this would have been okay to mention, like it definitely, and I didn't require, but I think it would have been good to mention that, to say something like, it's worth noting that we expected red light and green light to have different results, but it turns out that they had the same results, which was unexpected. But what we're talking about here was that what we should have put was the errors in the experiment themselves, and we talked about them during the experiments. Do you remember what any of them are? Like what were some of the flaws in the way we performed these experiments?
I know that my partners, whenever we were putting the cuvettes under the lamp, we didn't pull them out exactly at the same time, so that's probably a buffer.
That's one of the flaws. Yep. Um, then there was the fact that you're eyeballing it, right? So we're not really looking at exact pHs. It's basically up to interpretation, where you're comparing one, just comparing the color of two different things and seeing which ones match. So that's an error. The fact that they were right next to each other. So, for example, something that was in the green light, I'm sure, was also getting a little bit of red light, right? Because they weren't in little boxes. They were also exposed to the other colors and just stuff like that. We talked about a few of them, but anyway, that's what you should have listed there, right? All the different errors, the flaws in the experiment, as opposed to the flaws in the results. And then this one here, you must have got it right. Now that we've looked at your lab, I don't remember exactly what you wrote, but I remember you had a good grade. So you must have answered this. This was a question in the lab, and you must have gotten it right. But so you're supposed to explain something else you could have done, some other way of testing your hypothesis. Because remember, in the scientific method, when you test your hypothesis, you're supposed to come up with a lot of different predictions in the real world as opposed to in lab. So you did this experiment, it supported your hypothesis, but in a perfect world, you would go on and do another experiment. So, for example, you could grow, I don't know, pea plants, and you could grow them in different colored lights, and you could measure them. Grow them for 6 months, and you measure them to see how tall they are every week, right? So that's a different experiment you could do. So instead of measuring the rate of photosynthesis by the pH for an hour and a half or however long it was, you could measure the rate of photosynthesis by plant growth over the course of 6 months or whatever it may be. Does that make sense?
Yeah, that makes sense.
All right. Here you didn't lose points for this, but almost everybody made this mistake. I just pointed it out. No one lost points again, but technically, yeah, table 4, that's in your lab, it's table 4, but in this lab report, that's table 1. And I'll say this to anybody watching this video. Hopefully, future students will watch this before they write their own lab report. You know, Anybody reading this lab report, this should make sense to them if they've never been in this class and if they've never read the lab, right? So if somebody were to read this lab report who's never read, has no idea what the lab is, they would hopefully look at this and say, "What?" That would probably confuse them. Like, well, where's table 1 through 3? But again, you didn't lose points for that. And then there was just an error right there, right? So when we go from 7.1 to 8.7, that's not a change of 1, because if it was from 7.1 to 8.1, that would be a change of 1. So that was one small point missed. And then no graph, right? So you lost points for not having a graph. So you got the figure, or you got the table, but didn't have the figure. Make sense?
Yeah, it makes sense.
All right. Well, that's it. Let me adjust your grade while I have you on here. Actually, let me stop recording because there's no need for people to deal with that in the video. And do you have any questions before I stop recording? Because this will be very good for, again, future students. Hopefully they'll watch these things before they do their own lab reports and they can learn from other people's mistakes.
Nope, you already answered all of them.
All right. Let's see. Let's stop recording.